Hi, I'm Young Joo Sun, a postdoctoral scholar in the Mahajan Lab. Today, I'll present our very recent publication in the Journal of Clinical Investigation on SARS-CoV-2 prophylaxis. Authors have no conflict of interest to declare. So as the COVID-19 pandemic began, we thought, how can we apply our expertise to help resolve the pandemic sooner? Our lab has been working on eye proteases, protein structure-based ocular disease pathology, and drug repositioning and repurposing. Therefore, we decided to look into the protein structures of SARS-CoV-2 and proteases involved in the virus pathology. Since everyone is kind of an expert in COVID-19, I'll briefly introduce the key pathologic features of SARS-CoV-2 and current treatments for them. So the SARS-CoV-2 has the spike protein called S protein, which is basically a current target for the antibodies generated by vaccine or in convalescent plasma. So the spike proteins are responsible for the entry of the virus to the human lung cells. So it first gets recognized by the human protease ACE2 and then processed by the human protease TMPRSS2, which cleaves the spike protein. Once the spike protein gets cleaved, then the spike protein can fuse to the human lung epithelial cells, and the virus can enter into the cell and release its viral components into the cells. However, one of the problems these days are that these spike proteins are where the mutations are found for the new SARS-CoV-2 variants. Thus, targeting the spike protein with the current therapeutics may lose its effects or power, or will keep in a question whether they will be efficacious to the future variants that has new mutations or even with the current variants. So we thought that, what will be the good target that will not be affected by the changes in the virus properties? And obviously it was a human protein. And one of the interesting thing was TMPRSS2 because it's a proteases that cut the spike protein. And the thing is that there were not that many treatments that was known to target the TMPRSS2 or this kind of a host protease. And in addition to that, the current drug repositioning strategies for the COVID-19 is mostly shotgun screening against the existing drug libraries. And most of the drugs identified so far are experimental compounds, so none of them can be fast repurposed for the clinical applications. So our goal here was to set whether we can find a drug that's tolerant to the SARS-CoV-2 mutations. And since the vaccines needs a very special administration techniques and the special facilities or instruments to distribute and store them for long term, we look for some drugs that can be administered orally and can be stored in room temperature. And also, not just the experimental compounds that people are looking at, we'd like to find something that's clinically proven to be safe. And also, we thought of what will be the drugs that can be used for the virus entry in general that can be used beyond this COVID-19 pandemic situation. From there, we have decided to target TMPRSS2. As I mentioned, TMPRSS2 is a human protease which has the catalytic S1 peptidase domain in it. When you look at the S1 peptidase family, the amino acid sequences at the catalytic site of them are quite conserved. Thus, it is very likely that the inhibitors targeting the other S1 peptidases may inhibit TMPRSS2 as well. So it makes the TMPRSS2 as a very good drug repositioning candidate. So we have created a novel drug repositioning computational algorithm called 3D phylofold, which can identify S1 peptidases which are similar to TMPRSS2 in terms of amino acid sequences and protein structure. As you can see on the left, the 3D phylofold algorithm has overlaid 78 S1 peptidase structures. Then it calculated their structural similarity and generated phylogenetic maps. When you compare the sequence-based tree to the structure-based tree, the proteins that are similar in sequence to TMPRSS2 tends to cluster near the structure-based tree as well. However, there are a few exceptions such as trypsin, which are sequentially distant from the TMPRSS2, but they are structurally closer to TMPRSS2. So based on this structural similarity, we have identified 6 proteases which has similar structure TMPRSS2, and the one like trypsin, this may be the one that might be missed by the conventional way of identifying similar proteases based on the sequence analysis. Based on the 3D phylofold result, we searched for the known inhibitors for the 6 proteases identified to be structurally similar to TMPRSS2. And we have identified 5 potential candidates from the search. Due to the time constraints, I'll skip to our animal data, but we have tested them in silico, in vitro biochemical assays, cellular autoproteolysis assays, cellular pseudovirus assays, and genuine SARS-CoV-2 virus cellular assays. Based on our biochemical and cellular data, we have decided to carry on an in vivo mouse experiment with Chemostat and Avoralstat. We used the humanized mouse model, which was the only available in vivo model at the moment. The infection phenotype of this mice was the SARS-CoV-2 virus in lung and loss of mice weight due to the COVID-19 sickness. We delivered our inhibitors to the mouse by intraperitoneal injection. After 4 hours, we challenged the mouse with the genuine SARS-CoV-2 virus. After the challenge, we treated inhibitor to mice 2 drug doses per day for 3 days. However, still, it is not clear how much virus is necessary to infect and show COVID-19 symptoms in human. Thus, we tested our drug's efficacy under 3 different viral dose challenged groups. The first group of mice was challenged with a viral titer of 10 to the power 4 PFU. The second group was with 10 to the power 5, and the third group of mice was challenged with the 10 to the 6 PFU per mouse. We could observe viral dose-dependent changes in drug effects, yet in every viral dose tested, we observed significant rescue effects from both Chemostat and Avoralstat. The viral titer in lung was significantly decreased after Chemostat and Avoralstat treatment compared to the control group. You can see here in the 10 to the 5th group and 10 to the 6th group. And for the mouse weight, we could see the mitigation of the mouse weight loss due to the COVID-19 sickness in every group upon drug treatment. Interestingly, in the group of mice challenged with the 10 to the power 5 viral titer, Avoralstat protected mice from the weight loss, while Chemostat did not protect the mouse from losing weight, but helped recover the weight faster than the control group. And in the group of mice challenged with the 10 to the power 6 PFU virus, even though both Chemostat and Avoralstat could not protect the mice from the weight loss, but yet the weight recovery was much more faster in Avoralstat treated groups compared to the Chemostat or the control groups. Thus, we could think of that the Avoralstat has a more superior effect than the Chemostat, and the Avoralstat can significantly reduce the SARS-CoV-2 infection in vivo mouse models. In conclusion, we have shown that TMPRSS2 is a good prophylactic target for COVID-19. We have developed a new novel drug repositioning strategy called 3D phylofold, and the drug that we identified, Avoralstat, can significantly reduce SARS-CoV-2 infection in mice. Our study is important because Avoralstat is an oral drug proven to be safe in phase 3 clinical trials for the hereditary angioedema. Thus, it can be fast repurposed for the SARS-CoV-2 prophylaxis in clinical trials. It also can be a good alternative and complementary prophylactic options to vaccine. Since Avoralstat targets host protein, it is very likely to be resistant to new COVID-19 variants because the mutations are at the virus, not at the human that we are targeting. Also, it can be applied to other future TMPRSS2 utilizing viruses. Historically, TMPRSS2 has been utilized by SARS, MERS, influenza A viruses such as H1N1, H3N2, and even in influenza B and such. Thus, whenever the new future virus comes out that utilizes TMPRSS2, we may use our drug as the drug to buy time until the vaccine or other effective drug candidates that's specific for the new virus comes out. Our drug repositioning strategy is very unique because it's hypothesis mechanism driven and it's also a structure-based method. So it is very likely that compared to the other big library-based shotgun screenings, we already know what's the action mechanism and what is the real interaction that will occur in the biological system to function. So it is more likely to be successful and expected to have the expected side effects on human body and the treatment of the diseases. And lastly, and surprisingly, our study is one of the first COVID-19 prophylactic studies based on the small molecular compounds that were validated in vivo prior to the clinical trials. I thank the lab members of the Mahajan Lab and the collaborators in U Iowa. The people who are the co-authors of the paper is highlighted in red. And I'd like to special thank to Dr. Goldberg and Christina Lusano in our department for letting me to work during the COVID-19 campus shutdown, and also the directors of MSKC and MCKC who allowed me to use their facility during the pandemic shutdown as well. Thank you.